Sperm, the male gamete. Sperm, also known as spermatozoa, are produced in the seminiferous tubules of the testes. They mature in the epididymis, where they can be stored for several weeks within the male reproductive system. But what happens after ejaculation? Once inside the female reproductive tract, the average lifespan of sperm is 24 to 72 hours. However, only the most motile and healthy sperm can survive for up to 3 days in cervical mucus. But the maximum fertilizing potential is within 24 to 48 hours. Clinical insight. Although millions of sperm are released, most die within a few hours. Only a small fraction reach the ampullary region of the fallopian tube where fertilization can occur. This limited survival time defines the male contribution to the fertile window. Ovum. The female gamete. The ovum, or more accurately the secondary oocyte, is released during ovulation, typically around day 14 of the menstrual cycle. It is arrested in metaphase 2 of meiosis and awaits fertilization. But here's the catch. The lifespan of the ovum after ovulation is just 12 to 24 hours. And even within that period, maximum fertilization potential exists in the first 12 hours. If not fertilized in time, the ovum undergoes atresia, a natural degenerative process, and is reabsorbed in the fallopian tube. Clinical insight. Because of this short viability, timing is critical. If the sperm doesn't meet the egg in this narrow window, fertilization will not occur. Summary table. Cell lifespan, optimal fertilization time. Sperm 24 to 72 hours, max up to 5 days. Best within 24 to 48 hours post-ejaculation. Ovum 12 to 24 hours, best within 12 hours after ovulation. Conclusion. Understanding the lifespan of sperm and ovum is key to predicting fertility. In natural conception, the ideal scenario is that sperm are already present in the reproductive tract before ovulation occurs, ready to fertilize the ovum as soon as it's released. This is why doctors and fertility experts focus so much on timing intercourse during the fertile window, which is typically 5 days before ovulation and the day of ovulation itself. 1. Fertilization, day 0. Fertilization is the very first step where life begins. A single sperm from the male meets an egg from the female, both carrying 23 chromosomes each. This fusion happens in the ampullary isthmic junction of the fallopian tube. The result: a zygote with 46 chromosomes, a full set for human being. At this moment, the baby's sex is determined, XX for a girl or XY for a boy. The egg immediately prevents entry of more sperm, a protective step called the cortical reaction. 2. Cleavage, days 1 to 3. Soon after fertilization, the zygote starts dividing, not growing, just splitting. First into 2 cells, then 4, then 8, and eventually 16 cells. This ball of cells is called a morula, which looks like a mulberry. It is still covered by a protective coat called the zona pellucida. By day 4, the morula enters the uterus. 3. Blastocyst formation, days 4 to 5. Next, fluid fills the morula and turns it into a blastocyst. Now the cells start to specialize. The outer layer, called the trophoblast, will become the placenta. The inner cell mass will become the embryo. At this point, the zona pellucida breaks away, allowing the blastocyst to prepare for implantation. Zona pellucida thins and ruptures called hatching. 4. Implantation, days 6 to 9. The blastocyst now attaches itself to the posterior wall of the uterus. This is called implantation. The trophoblast invades the uterine lining and splits into 2 layers. Cytotrophoblast, the inner layer. Syncytiotrophoblast, the outer aggressive layer that helps in attachment. By day 9, implantation is complete. Now the hormone HCG is released. This keeps the pregnancy going by maintaining the corpus luteum, which produces progesterone. 5. Gastrulation, week 3. In the 3rd week, a very important change happens. The embryo forms 3 germ layers from a flat sheet of cells. Ectoderm, which forms skin and the nervous system. Mesoderm, which forms muscles, bones, and the heart. endoderm, which becomes the gut, lungs, and liver. This process is called gastrulation, and it sets the stage for organ formation. 6. Neurulation, week 3 to 4. Soon after, the nervous system begins to develop. The notochord tells the ectoderm to form a neural plate, which folds into a neural tube. This tube will later become the brain and spinal cord. Some cells break away. These are neural crest cells, which become parts of the face, nerves, and more. 7. Placenta formation, weeks 2 to 12. The placenta is the baby's lifeline. It forms from both trophoblast cells and the mother's endometrium. Tiny finger-like projections called chorionic villi extend into the mother's blood. This allows exchange of nutrients, oxygen, waste, and hormones. By 12 weeks, the placenta is fully working. 8. Embryonic period, weeks 3 to 8. This is the most critical time, called the embryonic stage. All the major organs start forming. The embryo is highly sensitive to drugs, infections, and toxins at this point. By the end of week 8, the body starts taking on a human shape, with tiny limbs, face, and internal systems. 9. Fetal period, week 9 to birth. From week 9 onward, the baby is called a fetus. The body now focuses on growing and maturing. By around week 18 to 20, the mother feels movements. This is called quickening. The lungs are the last to mature, with important surfactant production beginning by week 24 and enough by week 34 to breathe air. 10. Amniotic sac and fluid. The baby grows inside a fluid-filled sac called the amniotic sac. The amniotic fluid cushions the baby, protects from infection, and helps lungs develop. If there's too little fluid, called oligohydramnios, it can be due to kidney problems. Too much fluid, polyhydramnios, may point to issues like blocked digestive tubes or brain defects. 11. Hormonal support during pregnancy. To keep pregnancy going, the body makes key hormones. In the first trimester, HCG keeps the corpus luteum alive, which makes progesterone. In later months, the placenta takes over and makes progesterone to maintain the uterus, estrogens for blood flow and growth, HPL to ensure energy for the baby. Conclusion. From a single-cell zygote to the birth of a baby, human development is an amazing step-by-step journey of life. Every stage is carefully timed, highly coordinated, and full of transformation. And it all starts with the union of two tiny cells that grow into a whole new human being. 12. Signals that initiate labor. Labor doesn't start suddenly. The baby and the mother's body work together to trigger it. As the uterus stretches near the end of pregnancy, it stimulates the baby's brain, specifically the fetal hypothalamus. This sends a message to the baby's adrenal glands to release cortisol, a stress hormone that matures organs and helps initiate labor. Meanwhile, in the mother's body, estrogen levels rise. Progesterone levels fall. This change increases the sensitivity of the uterus to a powerful hormone, oxytocin. Now both fetal and maternal oxytocin begin to rise. This causes strong uterine contractions, marking the start of labor. 13. The 3 stages of labor. Labor is a beautifully timed, natural process divided into 3 main stages. Small orange diamond stage 1. Cervical dilation. This is the longest phase, especially for first-time mothers. It begins with mild contractions and ends when the cervix is fully dilated to 10 centimeters. The latent phase. From 0 to 6 centimeters, slow and gentle. The active phase. From 6 to 10 centimeters. Faster and stronger contractions. This stage can last anywhere from 12 to 18 hours in a first-time pregnancy. Small orange diamond stage 2. Delivery of the baby, expulsion. Now the cervix is fully dilated. With strong uterine and abdominal muscle contractions, the baby is pushed out through the birth canal. This stage usually lasts 30 to 60 minutes. It ends with the joyful moment, the birth of the baby. Small orange diamond stage 3. Placental delivery. After the baby is born, the uterus continues to contract to expel the placenta, the organ that supported the baby throughout pregnancy. This happens within 5 to 30 minutes. If the placenta does not come out fully, it can cause dangerous bleeding, so this stage is carefully managed by doctors. 14. Postpartum changes. After birth. After childbirth, the mother's body begins to recover and reset. The uterus starts shrinking back to its original size. This is called uterine involution. A hormone called prolactin rises. This stimulates milk production for breastfeeding. For the next few weeks, there's lochia, a discharge of blood and tissue from the uterus. Hormones like estrogen and progesterone drop quickly, affecting mood, energy, and physical recovery. Development of zygote stage by stage. 1. Fertilisation, day 0. It all begins in the ampullary-isthmic junction of the fallopian tube. This is where the sperm meets the ovum. The sperm uses an acrosomal reaction to break through the corona radiata and the zona pellucida that surround the ovum. Once a sperm successfully enters, a cortical reaction takes place. This prevents other sperms from entering, protecting the egg from polyspermy. Next, the male and female pronuclei fuse, forming a single cell, diploid zygote with 46 chromosomes. This zygote is totipotent, meaning it has the potential to give rise to an entire human being along with supporting structures like the placenta. 2. Cleavage, days 1 to 3. The zygote now starts dividing, but without growing in size. This is called cleavage. Day 1, it divides into 2 cells. Day 2, 4 cells, then 8 cells. Day 3, reaches the 16-cell stage, now called a morula. Each cell is known as a blastomere. The zona pellucida still surrounds the dividing cells, preventing early implantation inside the fallopian tube. 3. Morula stage. Days 3 to 4. The morula, which resembles a mulberry, is a solid ball of 16 to 32 blastomeres. It starts moving toward the uterus, pushed gently by the cilia inside the fallopian tube. Here, outer cells start becoming more flattened, and inner cells rounder, marking the beginning of polarity or early specialization. 4. Blastocyst formation. Days 4 to 5. As the morula enters the uterus, fluid starts to enter the mass, forming a central cavity called the blastocele. This forms the blastocyst. The blastocyst now differentiates into 2 important layers. The trophoblast, the outer layer that will become the placenta, the inner cell mass, which will develop into the embryo. The zona pellucida begins to thin in preparation for implantation. 5. Hatching, end of day 5. Now comes a crucial step, hatching. The blastocyst sheds the zona pellucida, allowing it to come in direct contact with the uterus. This is essential for implantation. If hatching fails, implantation can't happen, leading to implantation failure or infertility. 6. Implantation, days 6 to 9. The hatched blastocyst now attaches itself to the posterior wall of the uterus, near the fundus. The trophoblast starts secreting enzymes to invade the endometrium. It further divides into cytotrophoblast, the inner cellular layer, syncytiotrophoblast, the outer invasive layer that breaks down maternal tissue and establishes lacunae. This is the beginning of placental formation. The syncytiotrophoblast also begins secreting HCG, a hormone that maintains the corpus luteum to support pregnancy. 7. Completion of implantation. By days 9 to 10. By day 9 or 10, the blastocyst is fully embedded into the endometrium. The surrounding uterine cells heal over it, enclosing it inside the lining. At this point, the decidual reaction begins. The endometrial stromal cells swell and store nutrients. This region becomes the decidua basalis, the maternal part of the placenta. Clinical insights. Ectopic pregnancy occurs if implantation happens outside the uterus, most commonly in the fallopian tube. In IVF, embryos are usually transferred at the blastocyst stage. Failure of zona hatching is a major cause of early implantation failure, sometimes requiring assisted hatching techniques. Timeline summary chart. Day stage key events. 0 fertilization zygote formed. 46 chromosomes. 1 cleavage begins 2 cell stage. 2 cleavage continues 4-8 cell stage. 3 morula 16 cell enters uterus. 4 to 5 blastocyst formation cavity plus differentiation. Trophoblast plus ICM. 5 hatching zona pellucida disappears. 6 to 9 implantation trophoblast invades endometrium. 9 to 10 implantation complete HCG begins. Decidualization of endometrium. Clinical correlations. Ectopic pregnancy. Implantation outside uterus, e.g. fallopian tube. IVF. In vitro fertilization embryos usually transferred at blastocyst stage. Implantation failure. Common cause of early pregnancy loss. Zona pellucida defect. May require assisted hatching in IVF. Zygote to blastocyst. Cell divisions and structural stages. 1. Zygote, 1 cell stage. Time. Day 0, within 24 hours of fertilization. Location. Ampulla of fallopian tube. Formed by fusion of sperm and secondary oocyte. Contains 46 chromosomes, diploid. Male and female pronuclei fuse. Surrounded by zona pellucida and vitelline membrane. Totipotent single cell. Capable of forming entire organism plus placenta. 2 cell stage. Time. 30 hours after fertilization. Process. First mitotic cleavage. Equal division into 2 identical blastomeres. Still within zona pellucida. No increase in overall size. Cleavage equals cell division without growth. Cleavage is holoblastic. Entire zygote divides. 3-4 cell stage. Time, 40-50 hours after fertilization. Each blastomere divides to form 4 smaller blastomeres. Cells begin to show asynchronous division. Not all divide at the same time. Still enclosed in zona pellucida. Prevents premature implantation. For 8 cell stage. Time day 2 to 3. More compaction begins. Blastomeres start adhering tightly due to ecadherin. Start of cell polarity. Differentiation potential begins to vary. Cells still totipotent or early pluripotent. 5. 16 cell stage, morula. Time, end of day 3, 72 hours. Structure name, morula, Latin mulberry. Appears like a solid ball of 16 to 32 cells. Enters uterine cavity via fallopian tube cilia. 2 cell groups. Outer cells become trophoblast. Inner cells become inner cell mass, ICM. Still surrounded by zona pellucida. 6. Early blastocyst, day 4 to 5. Time day 4.5. Key event. Formation of fluid-filled cavity, blastocele. Blastomeres pump sodium, water follows, fluid cavity forms inside morula. This creates a blastocyst. Trophoblast, outer layer. Will form placenta. Inner cell mass, embryoblast. Will form embryo. Blastocele. Fluid cavity. Zona pellucida begins to thin. 7. Late blastocyst, hatched. Time day 5 to 6. Key event. Hatching of blastocyst. Zona pellucida ruptures, allowing direct contact with endometrium. Blastocyst hatches and becomes ready for implantation. Increased expression of adhesion molecules, integrins, selectins, on trophoblast. 8. Implanting blastocyst, day 6 to 9. Site. Posterior wall of uterine fundus. Trophoblast adheres to endometrium, releases enzymes, invasion begins. Trophoblast differentiates into cytotrophoblast, mitotically active inner layer. Syncytiotrophoblast, outer invasive multinucleated mass. HCG secretion starts, maintains corpus luteum. Stage-wise summary table. Stage time after fertilization, cell count, structure, name, key features. Zygote day 0 1 zygote totipotent single cell. 2-cell stage 30Hs2. First mitotic cleavage. 4-cell stage 40-50Hs4. Equal blastomeres. 8-cell stage day 2 to 3-8. Start of compaction. 16-cell stage, day 3-16 morula solid mass. Early polarity. 32-cell stage, day 4-32 morula prepares for cavitation. 64 plus cells day 4 to 5 64 plus blastocyst blastocele forms. Hatched blastocyst day 5 to 6. Hatched blastocyst zona disappears. Implantation day 6 to 9. Implanting blastocyst trophoblast invasion. Clinical importance. Ectopic pregnancy. Implantation occurs outside uterus if zona pellucida ruptures prematurely. IVF protocols. Embryos are often transferred at blastocyst stage, day 5. Implantation failure. Can occur if zona does not rupture, hatching defect. Stem cell research. ICM of blastocyst is source of embryonic stem cells. Types of pregnancy, classified. 1. Based on location. A. Intrauterine pregnancy, normal. Fertilized ovum implants in uterine endometrium. Development proceeds normally. Most common and viable type. B. Ectopic pregnancy, abnormal location. Implantation outside uterine cavity. Dangerous and non-viable. Types of ectopic pregnancy. Site features. Tubal, most common 95%. In fallopian tube, ampulla, isthmus, fimbriae. Ovarian ovum implants on ovary. Cervical implants in cervical canal. Abdominal on peritoneal surface. Rare, high mortality. Cornual/interstitial at uterine tubal junction. High risk of rupture. 2. Based on number of fetuses. Type description. Singleton 1 embryo, most common. Twin pregnancy 2 embryos. Triplets etc. More than 2, higher order multiple pregnancy. Twins can be dizygotic, fraternal 2 separate ova, 2 sperm, genetically different. Monozygotic, identical 1 zygote splits. Genetically identical. 3. Based on viability. Type description. Viable pregnancy embryo/fetus can survive after 24 weeks. Non-viable pregnancy not capable of surviving, e.g., an embryonic pregnancy, molar pregnancy, ectopic. For, based on outcome. Type definition. Normal, term. Pregnancy reaches 37 to 40 weeks, ends in live birth. Miscarriage, spontaneous abortion. Pregnancy loss before 20 weeks. Stillbirth, fetal death after 20 weeks. Induced abortion, medically or surgically terminated pregnancy. Molar pregnancy, abnormal trophoblastic growth. Hydatidiform mole. 5 special types. Type description. Molar pregnancy, trophoblastic disease with grape-like swelling, can be complete or partial mole. Heterotopic pregnancy, simultaneous intrauterine plus ectopic pregnancy, rare. Chemical pregnancy, very early loss, before ultrasound detection. Blighted ovum, empty gestational sac. No embryo. Missed abortion, fetal death with retained products inside uterus. Threatened abortion, vaginal bleeding without cervical dilation. Inevitable, incomplete, complete abortion classified based on progression of pregnancy loss. Summary chart. Classification basis types. Location, intrauterine, ectopic. No of fetuses, singleton, twins, multiples. Viability, viable, non-viable. Outcome, normal, miscarriage, stillbirth, abortion. Special cases, molar, heterotopic, blighted ovum, missed, etc.